Acetyl-CoA is very essential for life, so detecting how much of it is available in the cell or cells, depending on which organism we are talking about, is very important and beneficial for us. We know that when acetyl-CoA is present in a protein known as 3OTW, it is stable and doesn't unfold. But once the acetyl-CoA is removed from the binding site of 3OTW, the protein unfolds. We can see here when comparing the apo version of 3OTW which doesn't contain acetyl-CoA to the holo version of 3OTW which does contain acetyl-CoA that the alpha helices and beta sheets are very unstable and relatively far apart from each other because the protein is unfolding. We can actually use this property of 3OTW to detect whether or not it contains acetyl-CoA. And this can be made possible with the help of FRET-based biosensors. So let me briefly explain what FRET-based biosensors are and how they work. So the example used here is of YFP, yellow fluorescent protein, and CFP, cyan fluorescent protein. CFP emits 490 nanometers of light and YFP gets excited at this exact wavelength. So a spectral overlap can occur and what we call a fret can occur where energy is transferred from CFP to YFP which causes YFP to glow and CFP to get dimmer. For example, if we attach CFP to protein Y and YFP to protein X, when they are more than 10 nanometers apart, they can't produce a fret. But once they are less than 10 nanometers apart, they can do this, where energy is transferred from CFP to YFP, making CFP become dimmer and YFP to glow. Now going back to 3OTW, if we attach CFP and YFP to different parts of it, then they would be too far apart to produce a fret in the unfolded version of 3OTW. But in the folded version, they would be less than 10 nanometers apart, allowing them to produce a fret. So if a fret is produced, we can understand that acetyl-CoA is present